Hi, I welcome you all to another lecture in Anthropology. In this lecture, we shall discuss Unit 9.4 Part D. Unit 9.4 Part D in Paper 1 Anthropology. We will be discussing genetic imprints in human disease, genetic counseling, human DNA profiling, and so on. First, we shall begin with genetic imprints in human disease. We shall begin with genetic imprints in human disease. What is the meaning of genetic imprinting? The differential expression of the gene based on the parent from whom the gene is inherited is called as genetic imprinting. I will repeat. The differential expression of the gene based on which parent we are inheriting it, based on which parent from whom the gene is inherited is called as genetic imprinting. I will explain this genetic imprinting taking the example of Huntington chorea. Huntington chorea. Huntington chorea is a neurodegenerative disorder where the person who is affected by this disease or who has this disease, he or she will make jerky body movements. Just like how you will see many people who are drunk during late night or evenings, you can find many people on the street, they will just keep dancing without their control when they're walking on the streets, isn't it? So like this, even these people who are suffering from Huntington's chorea, they will keep making jerky body movements, that is their hands, limbs, the legs, entire body parts will be just moving like this without their control. We will feel that possibly they are dancing, okay, they are drunk and they are dancing, but that is not the case. Actually, without their control, involuntarily, their body will be moving. So we have already discussed, and I even showed you a video of patients suffering from Huntington's chorea when we discussed pedigree analysis, right? Either when we discussed pedigree analysis or chromosomal aberrations, I showed you the video also on YouTube. So we know what Huntington's chorea is, right? So this is a disease, it is a neurodegenerative disease which people get at an older age, right? So the genes for this Huntington chorea disease can come either from the father or from the mother. If the gene for this disease comes from the father, then you will get the disease at the age of around 40 to 45 years. If you are getting the gene for this Huntington chorea disease from your father, then you will see the onset of this disease when you are around 40 to 45 years old. Okay, so if you are getting the genes for this disease from your mother, so if your mother's gene is responsible for the onset or in the formation of this disease in you, then you will get the disease at around 60 to 65 years of age, when you are 60 to 65 years old. See here, there won't be any differences in the disease. The symptoms of the disease will be the same. The severity of the disease will be the same. However, only the onset of the disease, that is at what age you will get this disease, only that varies. So if you are getting the gene from your father, you will get the disease at 40 to 45 years of age. If you are getting the gene for this disease from your mother, then you will get the disease at 60 to 65 years of age. So you might ask me, so why is it, ma'am, there is the severity of the disease is the same, the symptoms are the same, and the genes can come either from the father or from the mother, then why is it that if I inherit the disease from my father, I'm getting it at a very younger age, whereas if I'm inheriting it at a later, if I'm inheriting it from my mother, then I'm getting it at a later stage in life. Why is it so? The reason for this is differential methylation of DNA. It is because of differential differential methylation of DNA. What is DNA methylation? So during the process of formation of the sex cells, that is ova or ovum in females and the sperm in the males, isn't it? So in the male, the sex cell or the germ cell is sperm, and in the females it is ova or ovum. So during the formation of the sex cells, there is continuous cell division that is happening, isn't it? There is continuous cell division happening, and DNA replication also happens within these cells. So during the formation of ovum or during the process of formation of sperm, there is continuous DNA replication happening because of the continuous cell division which is happening. So during this process of DNA replication, the methyl group gets added to DNA. So DNA is made up of 4 nucleotide bases, isn't it? What are they? Adenine, guanine, cytosine and thymine, isn't it? What are they? Adenine, guanine, cytosine and thymine. Adenine always base pairs with thymine and guanine always base pairs with cytosine, isn't it? So what is DNA replication? DNA's replication is forming a duplicate or forming another exact copy of the existing DNA, right? So just like how we make a photocopy, right? If when we are preparing for examinations, suppose your friend has some good material and you want a copy of that, you will go to the photocopy or photostat place and you will make an exact copy of that. Similarly is the process of DNA replication. So once one DNA will be there, you make an exact copy of that. That is called as DNA replication. And in detail about DNA replication, we discussed in the very first class of physical anthropology, right? So I hope you remember that. So during this process of DNA replication, a methyl group means CH3. This is the methyl group, right? Methyl group gets added to cytosine. Okay, this process is called as DNA methylation. This process is called as DNA methylation. Okay, the addition of methyl group on cytosine by the enzyme. So this DNA methylation process is carried out by an enzyme called as DNA methylase. Okay, so this methyl group is added to cytosine by the help or by the aid of this DNA methylase enzyme, and this process is called as DNA methylation. Okay, in simple terms I tell you, the addition of a methyl group to the DNA is called as DNA methylation, and it is carried out by the enzyme DNA methylase. Okay, did you understand what is DNA methylation? The addition of a methyl group by DNA, by the enzyme DNA methylase to the cytosine of the DNA is called as DNA methylation. So what is the function of this DNA methylation? DNA methylation controls the gene expression. Okay, the important, what is the use of this? What is the function of DNA methylation is, it controls the gene expression, that is when and how the gene should be expressed. That will be controlled by the addition of this methyl group to the DNA. Okay, so this is still an emerging science and lot of research is going on in this. So do not worry much about what is DNA methylation. See, just understand that the what is genetic imprinting. Okay, so the differential expression of a gene based on the parent from which the gene is inherited is called as genetic imprinting or genomic imprinting. And why is there a difference between the onset of the age when the gene expresses depending on the parent from whom the gene is inherited? That is because of the process called as DNA methylation. DNA methylation is a process by which a methyl group here means CH3, the methyl group is added to the cytosine of DNA during the process of DNA replication, which happens during the formation of the sex cells, which is the sperm in males and ovum in females. So this process of DNA methylation is facilitated or it is carried out by an enzyme called as DNA methylase. This entire process is called as DNA methylation, and it is this DNA methylation which controls the expression of the gene. Okay, it controls the expression of the gene, and that is why this process is responsible for the difference in the age at which you get the disease depending upon which parent you are inheriting that gene for the disease. Okay, so it is DNA methylation, that is differential DNA methylation. The differential methylation which happens in the sperm and the ovum cells is responsible for the difference in the age at which you get the disease. So when you inherit it from your father, you get it at an earlier age. When you inherit it from the mother, you get it at a later stage in life. Okay, did you understand so much so far? What is genetic imprinting and what is differential expression? What do we mean by DNA methylation and the enzyme which causes it? Now we shall discuss few characteristics of DNA methylation. Sorry, we shall discuss few characteristics of genome imprinting. See, genome imprinting and genetic imprinting both are the same. So do not get confused in your exam. If suppose instead of genetic imprinting, they have mentioned the words genome imprinting, do not think that is some other new topic which you have not prepared. Okay, genome imprinting and genetic imprinting both are the same. So once again, I will tell you what is genome imprinting. So during the passage of the genes from the parents to the offsprings, the autosomes and the X chromosomes are printed separately. That is very clearly whether the genes are coming from the father or whether the genes are coming from the mother. They will have a print on that. See, print means you have to understand it is not literally taking some stamp or ink and printing it. They have that identity. Okay, so during the passage from the male and the female parents, the autosomes, we know what is autosomes, right? Right? Autosomes, allosomes, we've discussed all this in the very first class. So, during the passage of the genes from the parents to the offsprings, what happens is the autosomes and the X chromosomes are stamped differently. Okay, so when the autosomes and X chromosomes are coming from the father, they will be stamped differently, and when they're coming from the mother, they will be stamped differently, so that they will bear the marking of which parent they are coming from. Okay, and similar, though these two genes are similar genetically, their expression, that is their phenotypic expression, varies. So see, I will give you the example of this Huntington's chorea itself. Say this is a gene, say this is a gene for Huntington's chorea which is coming from your father, and this is a gene which is coming from your mother for the same disease, Huntington's chorea. To look at both the genes, they will look exactly identical. There won't be any difference. Okay, that is genetically, that is whether they have A, T, C, G, C, G. Here also they will have A, T, C, G, C, G. Okay, are you understanding what I'm trying to tell? They will not have only these many base pairs. It will be a huge complex thing. Okay, but what I'm telling you is genotypically, that is at the genetic makeup level, if you see, both of them will look same, whereas their expression differs. So when it comes from the father, the expression will happen at a very younger age, whereas when it comes from the mother, the expression happens at a later stage. So based on that, we can identify whether the gene is coming from the father or from the mother, right? So again, one more thing which you have to keep in mind is not all genes have this marking. Okay, genetic imprinting is not found in all the genes which we inherit from our parents. Only in few genes and few diseases we have discovered it so far. That is why I told you that is still it is an emerging science and lot of research is still going on in this field. Okay, now we shall discuss few characteristics of genetic imprinting. I hope you all are following, isn't it? It's very simple, don't worry. Characteristics of genetic imprinting or genome imprinting. Characteristics of genetic imprinting or genome imprinting. First one is the imprinted genomes express variously. The imprinted genomes express variously. Imprinted genomes express variously. So, the genes can have a different magnitude of expression. That is, one gene might see the gene for certain disease. As I told you the example of Huntington chorea, if you inherit from your father, you will get it at an earlier age in life. If you inherit it from your mother, then you will get it at a later stage in life. Similarly, one will express earlier and one might express at a later stage in life, and one gene might have some enhancing effect on some function, or another one might have some retarding effect on the same function. So the imprinted genomes express variously. Okay, so as I already told you, though both the genes are same at the genetic level, their expression differs. That is, when it comes from father, you get at an earlier age. When it comes from mother, you get at a later stage. Similarly, there will be other genes which have genetic imprinting. So those genes, if you inherit from your father, it might help in the functioning of some activity, whereas if you are inheriting that gene from your mother, it might inhibit that activity. So they can be opposing functions also. That is why we say the imprinted genomes express variously. Next, the genomic imprints are erasable. So genomic imprinting is not a permanent modification of the gene. Keep this in mind. Genomic imprinting is not a permanent modification of the gene. So whatever genomic imprints you have received from your father and your mother, it will not be transferred from your children to the next generation. Okay, so what happens is whatever imprints of the genes that you have received from your father and the mother, when your son or your daughter in their body, when the sex cells are formed, these imprints will be erased. So there is no chance of these imprints being carried from one generation to another, and therefore this is not a permanent modification of the gene. So those imprints will be carried only for the next generation, that is from you to your children. From your children it will not be carried over to the future generations. Did you understand? So genetic imprints are erasable. So it persists only for one generation, and it is when the child, whether your son or daughter, when the sperms or the ovum are formed in their body, that time those imprints will not be carried forward from them to their children. That time new imprints will be formed. So genetic imprints are erasable, and these are not permanent modifications of the gene. Next, gene imprinting is most common in mammals and flowering plants. It is most common in mammals and flowering plants. Okay, so you can find this process of genetic imprinting very commonly in mammals and flowering plants. Gene imprint is species specific. Is species specific. It is genetic imprinting is species specific. Genetic imprinting is species specific. So a gene imprinted in one species may not be imprinted in another species. Okay, so say there are certain genes which are imprinted in human beings. Those, it is not necessary that those specific genes will be imprinted in another mammal, say a dog or a cat or any other animal. So in those animals some other genes might be imprinted. Okay, so the imprinting of the genes varies from species to species. Whatever genes are imprinted in humans need not be imprinted in other animals. So whatever genes are imprinted in the other animals, say rat or cat or dog or mouse, whatever genes are imprinted in those organisms need not be imprinted in us human beings. So it differs from species to species. Say for example, there is a gene for insulin like growth factor 2. There is a growth factor 2. There is a growth factor 2. So there is a hormone or a growth factor 2 which is present in the mouse or the mice. So in that gene you can find imprinting happen in the mice. Whereas in human beings we cannot find the imprinting happening in insulin, which is a similar growth hormone. Insulin is not a growth hormone, which is also a hormone. So the function of this growth factor 2 is similar to insulin in mice. But are you following what I am trying to tell you? See, whatever genes are imprinted in us, it is not necessary that those same genes coding for those characters only will be imprinted in a mouse or in a dog or in a cat. There will be some genes which are imprinted in a cat. It is not necessary that those same genes will be imprinted in us human beings also. So depending on the species, so say for example, let us take ABCD to make things simple. Suppose there is a gene A, B. Suppose we think, let's take it as A, B, C, D, E. So okay, suppose gene A is being imprinted in human beings. In mice, some gene C will be imprinted. In dogs, some E will be imprinted. Okay, and in cats, some B and C will be imprinted. So, depending on the species, different genes will have imprinting. And it is not necessary that if one gene is imprinted in a human being, in all other organisms or all other mammals also, the same gene should be imprinted. And it varies from animal to animal, mammal to mammal, species to species. This is what I am trying to tell you. Did you understand? So there is this growth factor 2, which is being imprinted in mice. Okay, we have found that this gene which is coding for growth factor 2 in mice has been genetically imprinted, whereas that is not the case in human beings. Okay, so there is a difference. So the genetic imprinting is species specific. Then genetic imprinting is implicated in many diseases. Okay, you can find genetic imprinting in many diseases, like you can find it in many genetic diseases can be found in genetic imprinting can be found in many diseases like cancer of several forms, Angelman syndrome, and all these things. Fine. Then genetic imprinting is not a rule. Keep this in mind. Genetic imprinting is not a rule. What do you mean by this? Suppose there are thousands, some 10,000 genes in our human body. It is not necessary that all those 10,000 genes have to be genetically imprinted. So when the offspring, okay, when a parent to an offspring, when an offspring gets certain genes from the father and the mother, it's need not necessary that all the genes will show difference, saying this is the genes which you have got from your father and this is the genes which you have got from your mother. It is not necessary. There will be certain genes which are imprinted. There will be certain genes which are not imprinted. So genetic imprinting of the genes is not a must. It is not a rule. Did you all understand this? These are the few characteristics of genetic imprinting or genomic imprinting. First and foremost, the imprinted genes express variously. So the gene, whether you are inheriting it from your father or your mother, it will show differential expression. So the time of expression may vary, or the way of expression might also vary. So one gene, whether if you are inheriting it from your father or mother, might suppress a reaction or a function, whereas from the other parent when you are inheriting, it might not suppress the reaction. In fact, it might help the reaction to go, or help the function to progress successfully. So that way also it can vary, and it also varies in the onset of the disease, that is the age at which the onset of the disease happens. Next, the genomic imprints are erasable. So these are not permanent modifications of the gene. So those imprints, whatever you have inherited from your parents, will not be transferred from you to your children. Okay, so when your child, when in your body, whether you're a boy or a girl, a sperm or a ova is produced, those imprints which you have got from your father or your mother will not be carried to your sperm or your ovum cell. So those will not be carried to your next generation. So these are erasable, that is genetic imprints are erasable, and it will not be carried across generation. It is only carried from parents to offspring, only for one generation. Okay, then genetic imprinting is most common in mammals and in plants. And then genetic imprinting is species specific. Okay, so those genes which are imprinted in me, that is a human being, might not be imprinted in a cat or a dog. And those genes which are imprinted in a cat might not be imprinted in a dog. Okay, so the genetic imprinting varies and it differs from species to species. And then genetic imprinting can be seen in many diseases like cancer, embryonic tumors, Angelman syndrome, Huntington's chorea, like this. And then gene imprinting is not a rule. So no matter how many ever thousands of genes you have in your body, it is not a rule that all of those genes will be imprinted. From looking at those genes, it is not necessary that all of these genes will carry a stamping to say whether you have inherited that from your father or your mother. There are certain genes which express the stamping and other genes which do not show that. So genetic imprinting is not a rule, and it is not found in all the genes which you have in your body. Okay, so this genetic imprinting is still a new science. So a lot of research is happening in this sector. Then the direct modification of the DNA through DNA methylation is responsible for the differential expression of the DNA, differential expression of the gene. So when a child receives chromosomes from its father and mother, the paternal and maternal chromosomes bear their own imprints. So in the gonad of the offspring, the parental imprints are erased. As I already told you, whatever imprints you have got from your father and mother in those genes, you when there is the formation of gonads in you, that is in your body, when if you're a boy, sperm cells, or if you're a girl, ovum cells, when they are formed, those imprints will not be carried in that, so it will not be transferred to the future generations. So your gonad during the formation of the sperm or the ovum cells will erase those imprints, okay, and it will not let it to be carried to the future generations. And then the genes coming from the mother in most cases is more methylated. Okay, the genes coming from mother is having more methylation in comparison to the genes coming from the father, and the methylation pattern of the gene changes when the gene travels from the mother to a son. So I told you why there is a difference in the onset of the disease when you are getting the gene from your father or your mother. If you are getting the disease because of the genes that are coming from your mother, you will get the disease at a very later stage. I told you in the example of Huntington's chorea, it is around 60 to 65 years of age. Why is it so? Because when the gene is coming from the mother, it will be more methylated. Okay, more methylation prevents expression of the gene at a very earlier age. Because there is more methylation, it does not express at a very earlier age, and it expresses at a very later age, and that is why you are getting the disease at a later age. Whereas the gene coming from your father will have less methylation, as a result of which the gene expresses at a very earlier age, and that is why you are getting the disease at a earlier stage in life. Okay, then a son receives highly methylated DNA from his mother, but when he passes on his genes to his offspring, it will be less methylated. Okay, so did you understand genetic imprints in human disease? The differential expression of DNA based on which parent the gene is coming from is called as genetic imprinting. That is, whether you are inheriting the gene from your father or are you inheriting the gene from your mother. If you are inheriting the gene from your mother, then the DNA will be more methylated, as a result of which the gene gets expressed at a later stage in life. That is why you get the disease at a later stage. Whereas if you are inheriting the gene from your mother, from your father, the DNA or the gene which you are inheriting from your father is less methylated. So because of less methylation, the gene expresses at an earlier stage. So I already told you the process of DNA methylation, right? So the addition of methyl groups to the DNA during the process of DNA replication while the formation of the sex cells is called as DNA methylation, and DNA methylation actually controls the gene expression. If a DNA is more methylated, the DNA will express at a very later stage in life. If it is less methylated, it will express at an earlier stage in life. That is why when you inherit from your father, you are getting the Huntington's chorea at an earlier stage of around 40 to 45 years. When you are getting the gene from your mother, because there is more methylation of the DNA, you are getting the disease at 60 to 65 years of age, because the expression of the gene takes longer time because of excessive methylation, and it is this methylation of the DNA which controls when the gene will get expressed. If more methylation is there, gene will express at a later time. If less methylation is there, it will express at an earlier age. Did you all understand this much? Fine. So this is about genetic imprinting or genome imprinting in human disease, and we also discussed the characteristics of genetic imprinting. This is a very new science, okay? It is still a lot of research is happening in this. So if you just understand the simple understanding, whatever I explained to you, that is more than sufficient. Fine. Now we shall move on to the next topic, genetic screening.